We will now begin the fourth technical session on aquatic animal diseases, diagnostics, prophylaxis, and therapeutics. This session will be chaired by Professor Indrani Karunasagar ma'am and Dr. Basanta Kumar sir. Please come to the dais, sir. Ma'am. Yes, sir. This session is coordinated by Dr. Sujit Kumar from SIBA and Dr. Anutosh from NBFGR. I now invite the coordinators to comment the session. Today's keynote speaker is Hatnang Dong and lead presentation by Dr. K.V. Rajendran sir. I request
Thank you, madam. Very good morning to all. So, as ma'am mentioned, the session will be chaired by Professor Dr. Indrani Karunasagar. So, although we all know her achievement in the field of aquatic animal health, so I'll just read a few. Professor Dr. Indrani Karunasagar currently serving as Director of Projects and the DST Technology Enabled Center at NIT University, Advisory Board Member of UNESCO International Decade of Sciences for Sustainable Development, Head of the FAO Reference Center for Antimicrobial Resistance and Aquaculture Biosecurity, and expert member on antimicrobial use at the OA. She has held postdoctoral and visiting scientist role at leading global institutions such as University of Stirling, University of Maryland, University of Wurzburg, and Ghent. Professor Karunasagar has received numerous prestigious awards, including National Technology Award from Government of India DBT, Sri M. Visweswaraya Lifetime Achievement Award from Government of Karnataka, and the prestigious Rafi Ahmed Kidwai Award of ICR, and also Outstanding Women Scientist Award from ICR. With over 400 scientific publications and over 14,000 citations, she continues to shape the future of aquatic animal health in the country as well as globally. So I welcome Madam to the session and for the further proceedings. I request Dr. Sujit to introduce Dr. Kidas sir.
Good morning to you all. I am delighted to introduce our chairperson, Dr. Basant Kumar Das. Dr. Basant Kumar Das is an accomplished scientist and administrator. He is currently serving as a director of Central Inland Fisheries Research Institute, Seafree, Barrackpur. He holds a PhD from Orissa University of Agriculture and Technology and has completed 2 postdoctoral fellowships at FRS Marine Lab, Aberdeen, UK, and Barnes College of Engineering, University of California, USA. With over 30 years of research experience, Dr. Das has led numerous projects in biomolecular characterization, proteomics, bioinformatics, biosensor diagnostics, monoclonal antibody development, cellular biology, and microscopy field. His prolific academic output includes more than 500 research papers, 40 book chapters, 8 patents, 13 copyrights, 7 trademarks, 4 registered designs. Recognized nationally and internationally, he has received awards such as Lal Bahadur Shastri Young Scientist Award, Rafi Ahmed Kidwai Award, DBT Overseas Associateship, Best Scientist, Best Speaker, and Lifetime Achievement Award. His leadership and collaborative skills have significantly strengthened India's inland fisheries research and global scientific partnerships. Thank you, sir. Thank you for chairing the session. Now I hand over the sessions to the chair. Thank you.
A very good morning to one and all here. I'm sure the hall will get full in the next few minutes because there is a very exciting session awaiting all of us. At the outset, I thank Dr. Kuldeep Lal for having invited and to let us all come together. All the doyens in the field are here, and it's exciting to see very young people participate, and the future of the diseases in Asian aquaculture is in the hands of those young candidates, young scientists who are out here to get the best from all of the seniors here. It is my privilege and pleasure to introduce the most important speaker for the day, Dr. Ha Thang Dong. He is recognized, yes, as an aquatic health researcher and therefore there to give this address. He has specialized in tropical aquaculture diseases, diagnostics and vaccine development, as the session name itself indicates. And his pioneering work spans host pathogen interactions, sustainable disease control strategies, and nanobubble enhanced vaccine technologies, a need of the hour for better administration of vaccine. With over 110 peer-reviewed publications, he serves on the advisory board of leading journal and is a consultant for FAO as well as World Fish. Based at the Asian Institute of Technology, he holds an adjunct professorship also at James Cook University, Singapore, and we are more than delighted to have him begin an exciting day by giving his opening talk now. Thank you all very much. Thank you, Dr. Dong.
Oh, I haven't seen my slide here. No slide in this area. Okay. Good morning, everyone. It's I'm truly honored to have a chance to be here, to meet many colleagues, friends, and also very new face. It's always I feel always very special every time I come back to India. 3 months ago, my wife and I have a couple of weeks travel to northern India, and we have freedom from work, no email, no pressures, and then we talked to each other. One day that we came up with a question: can we do good science without sacrificing animal? Yeah, because like you, I'm a pathologist. I'm working with diseases. And many of my work involved in animals. And sometimes we need to kill hundreds to thousands of animals to be able to understand something. So that's why we came up with this question. And we make a decision that we turn our direction to be able to understand or make a sense in another way. I don't know how it's going to be, but at least we need to dedicate for making some new knowledge by challenging ourselves in the area that we're not familiar with. So my talk today is about non-lethal methodologies in aquatic animal health research. And my outlines of my presentation today, I will start with a very brief introduction why we come up with this one, and then how non-lethal methods can be applied in different fields. For example, diagnostic, serosurveillance, immunological assessment, and microbiome pathobiome study, and also some other alternatives to animal, other approach that alternative to animals, and how these non-lethal methods align with recent global finfish health priorities. And then the final remarks. Okay, you may see this picture. These are created based on my imagination. So I asked ChatGPT, could you help me to illustrate this one in the picture? Okay. So this one is just, to seeing ourselves in animal place. So when we see ourselves in other place, animal place, we will be able to understand deeper. That own life being have values, and we should not. When we do, we need to respect and with responsibility. So, as I mentioned, like many of you, I use animal for research. And millions of aquatic animals have been using to certify for our experiment with the aim to understand, to reduce disease in aquaculture. So the study to improve animal health, but the fact is often come up with costing the life, right? We try to protect life, but we take the life from others. And the protecting life means that we need to sacrifice others. That's the question challenging us how we can do science in better ways. And I hope that we still can pursue a good science, but respecting animal life. And non-lethal approach is not something new. Many of you have been using, and people have using for many years, but we haven't make it empower the techniques and apply it widely to replace the animal. So non-lethal, sometimes like we use blood, we use mucus, fin clips. For shrimp, we use hemolymph, swimming legs, we use fecal sample. And even we use environmental nucleic acids from the water body when they keep the animal for diagnostic and also for other study. And we work with infected diseases, and we understand that many infected diseases in aquaculture are systematic infection. So with those disease, systematic infection, you can detect for diagnostic, non-lethal sample and lethal sample usually have the same value because the virus or the infected agent, they distribute widely. So you can get the fin clip, you can get the blood, and the result is the same that you get from liver and kidney. From my experience and also from other research, fin clip and blood appear to be the best non-lethal sample. That's what I understand now. And the understanding of tissue tropism, we work with diseases. We need to understand what the tissue tropism, what the setting pattern of the pathogen. If we understand that one, it can help us to get the right sample type for diagnostic. And yesterday, someone also reminded that, for diagnostic, sometimes we don't need to go with high tech. We need to go with very low tech and simple, very basic. And for some pathogen diseases, parasitic diseases, and fungal diseases, microscope examination of wet mouth, of stem smear, that enough for you to give some rapid diagnostic and what you can go further. And one of the roles of diagnostic is we try to target, to detect a symptomatic infection. It's in early detection when the pathogen load is very low. In that case, non-lethal sample, if we go together with high-tech, like qPCR or digital droplet PCR, the method that can detect to very low level of infection usually give very good result. So I would like to showcase to you some example that we have done earlier. So we work with several emerging diseases in aquaculture. And this is a couple of viruses that we work in Bramandi or Asian sea bass. The first one is scale drop disease virus. One of my master students working to compare between lethal and non-lethal methods, and we found that blood, mucus, and ectoparasites, the parasite that we taken from the fish, also have the value for detection. And blood appear to be the best organ for detection. It is the same result as other tissue. So blood is easy to collect, and we don't need to certify the animal. And also another study with another virus, we call Lactate Cancer herpes virus, or LCHV. This is the emerging virus of Asian sea bass. So when we develop the detection method for this one, and we also compare different types of sample, and it appears that fin appear to be the best one for diagnostic. Yeah, so we never think that, fin is even better than internal organ. And we also developed the diagnostic non-lethal methods for TIOV in tilapia. So one of my students compare different sample size with using blood and using liver biopsy. So we just get, the fish show you the liver position and the, the spot that we get the liver biopsy. And the result came out at blood have better detection result compared to liver biopsy. Okay. So all the systematic infection blood should be normally is a very good organ sample type for diagnostic. And we also explore nucleic acid, environmental nucleic acid that we get from the water. We just concentrate the water and extract for RNAs and use the qPCR method to detect the TIOV. And this is the work that we, very initial stage that we try to detect from water. It's not easy for virus, but we managed to make it happen. And we look into viral load of TIOV in water, where disease outbreak, and where no disease outbreak, but infection exists. And you see that we can quantify the amount of virus in the water. So with disease outbreak, the amount of virus is higher compared to the one with infection with no disease. So it means that if we do more data, we make a big data, we should be able to set the thresholds. The threshold that we be able to say that, okay, with this level, in the next 5 days, next 10 days, you may have the outbreaks. Yeah, so we haven't do enough. But this very simple work, but it's open, some new direction that is something possible. And nowadays, I think the farmer, they don't want to see that what happened with the fish, but they want to see that, can you forecast what's going to happen with my fish? Okay, so I would like to move to the second application is the use of non-lethal methods for serosurveillance. So serosurveillance is something not new as well. It has been using for other animal sector a lot, but not many dominant in aquaculture. So the basic serosurveillance is the way that we use the blood sample and to detect the presence of antibody against particular pathogens of interest, okay? And normally it can detect the exposure current infection or past infection. So current infection or past infection, if the fish develop immunity against the pathogen, we should be able to detect the antibody in the blood. And it's used to monitor the spread of the pathogens in the population. And also assess the vaccine effectiveness. That is, many people work with vaccine, and we know that this is very useful. And also understand natural immunity. Okay, so natural immunity is we haven't focused a lot, but that is something we should focus. And serosurveillance also used for genetic breeding program. Many companies they work with genetic breeding. They need to have we need to have the SPF broodstock. So serosurveillance, we have them to identify the one is naive, is not yet exposed to pathogens, and can be used at the broodstock. And this also can be, monitor their population as well to ensure that they are not exposed to pathogens. And for serosurveillance, normally we go with ELISA. The technique is commonly we use with ELISA, and it requires very small amounts of blood and without killing animals. So for one ELISA reaction, you may need only 1 microliter of blood. 1 microliter of serum, sorry. So I would like to showcase to you the result that we work with serosurveillance. Yesterday, I remember someone mentioned about multiple infection. That is the reality in aquaculture. Disease in aquaculture, we never see a single infection. So that is something that I did in my PhD work and public work that many people, also pay attention on that one. So at that time, we found that one fish may carry 2 to 5 pathogens in their body when disease outbreak. So come back to this one. Another way we also can be able to imply what pathogen they infect with. So we do the serosurveillance for 5 pathogens from 4 tilapia populations across 2 seasons, hot season and cold season. So each population, we get sample size of 10 fish, get the serum, and do detection, ELISA detection against 5 pathogens. The first one is Pseudomonas orthitis, Aeromonas supertii, Tilapia neck virus, Staphylococcus aureus, serotype 1A and 3. So the result is very interesting. So you see that each fish exposed to some, exposed to 5 pathogens. They have antibody against both all 5 pathogens. And majority of them, the positive sample, with higher prevalence in the cool season compared to hot season. So we have more disease in hot season, but seem like the antibody response and the seropositive and high level at the cold season. So there's something with, I don't know, there's something with the fish immunity or something with the pathogen profile in the water. Yeah, or combination of both. And it's interestingly that we found that there are 2 important pathogens, is TIOV and Streptococcus aureus, appear to be higher in cool season. Okay, the disease outbreak in hot season, but serosurveillance, the 1A is higher in Streptococcus aureus in cold season. Okay, so the data like this help us to understand, what our fish exposed to, what the system, in our system, what major pathogen we have in our system. And I also have some recommendation for serosurveillance. So when we currently, not many people using it, but in the future, I think that we should develop the serosurveillance toolkit that be able to target to a panel of pathogen, important pathogen in tilapia or other animals, and promote the use of serosurveillance in monitoring and for selective breeding program, and further explore the application, the use of mucosal antibody for serosurveillance. Why I come up with mucosal antibody for serosurveillance? From our experience when we work with vaccine, and we look for antibody response in both systemic and mucosal system. And there's something very interesting that, even antibody level in the mucus is significantly lower compared in the serum, but the pattern of antibody response you see is exactly the same, very, very the same. And happen to many pathogens. It's not all, but many pathogens they have. We can see this one. It means that the mucosal sample is very useful. We don't need to get the blood, we just take a swab, and then we can do ELISA, and we know, what we have. And another approach that we can use is the use of non-lethal methods for immunological assessment. This one is very useful for those working with vaccine, working with immunostimulant, working with probiotics. So, the fish immune system, when we look, we have innate and adaptive, and we have humoral and cellular response. So what we look for, we can classify to parameter for humoral response and for cellular response. So from humoral response, for example, like you may normally look into cytokine response, lysozyme, antimicrobial peptides. We look into complement protein pattern. We also look into antibodies and IgM, normally in systemic and mucosal, and IgT, mainly in mucosal system, and IgD as well. Or we look into cortisol level. That implies that how stressful our animals are. And also histoprotein and other blood chemistry. So when you go to the hospital, doctor take the blood and come up with a list of blood chemistry and give you the advice. So can we do this for aquaculture as well? Yeah. And also for cellular components that we look into leukocyte count can be total count or different, or you classify to different types of phagocytic activities, that how ability to engulf the pathogens, and respiratory burst is ability to kill the pathogens, or cytotoxic activity that we look for natural killer cells. In human, when you go to hospital, doctor can give you the test and how active of the natural killer cells. And also we can do for gene expression and marker, some biomarker as well. So what the current practice? Like many of you, I started with immune gene expression. I use qPCR, and that one become very simple and easy for me now. Every student in my lab, they can do it. But the problem is, okay, that is needle. Every time you collect sample, every time point that you need to certify animals, you need to collect the spleen, kidney, and intestine to look for gene expressions. And the mRNA level is not always reflect the level of protein. So sometimes RNA level is high, but they're not producing protein. So that's why the better approach that we look for protein. We measure the protein response instead of measure gene response, mRNA. And so currently there are several ELISA kits, commercial kit has been used. So I'm lucky that I have a project that's working with vaccine, and I have a lot of money to do it. And I feel that, oh, I don't want to do gene expression anymore, and write to funding agency that can I change to ELISA to measure protein level. And luckily that they approved and they support me, and I started to explore some commercial kit available. But even they're available on the market, but there's no data. You need to buy the kit, and you need to start to set up and understand whether the kits work for the purpose or not, whether it's work for the animal or not, because they develop maybe from different species, but then you adapt to the species of the interest. It may have a lot of challenge. But luckily that my students also here today, they're very active, and they'll be able to set up the many kits available, work for tilapia and Asian sea bass, two species that I'm focused on. And also many functional assays that we can be used to replay for challenge tests to be able to understand better. For example, bactericidal assay that help to see whether antibody or serum, we're able to combine protein in the serum able to kill the pathogen or not. And neutralizing assay, how antibody block infections. That is good for viral vaccine development. And agglutination assay, that is some lab, if you don't have the ELISA system, you can come back to very traditional approach that agglutination test is still working very well. And nitrozyme activity, so this one I also found that it's very useful when we work with different kind of vaccine. Sometimes we look for antibody, but we don't see antibody, but other components also very valuable, and it's help to save the fish from infection. And the need is currently we don't have many focus on how to measure cell-mediated immunity. So we can measure antibody easily, but the immune system of the fish, they have two arms of immune system. This one related to B cell, produce antibody, the other related to T cell, and is to kill infected cell. So sometimes when we work with vaccine with intracellular pathogen, they not activate the antibody response, but when we challenge, we see the protection very well. So that is something with another arm of immune system, but currently we don't really have the system to measure it. The next one, and if anyone work with microbiome, we should also work pathobiome as well. So people talk a lot about microbiome, but pathobiome is not many people talking about. Okay, and when we work with animal health, we need to understand the interaction. So microbiome is a collective of microorganisms present on and in animals that help to support on health and normal functions. So you can see it from the surface, you can see it from the gut, and even inside systemic system as well. And pathobiome is a subset of harmful microbes associated with diseases. Okay, so whether we can use, now the next-gen sequencing come and the price is also very cheap. And what we understand, we try to understand the balance of microbe in our system. And what kind of sample type is useful. Okay, and so currently I know that the majority of work with microbiome, they just grab the mucus or they collect the gut. This is a needle, they just collect the gut and look into microbiome and not really focus on pathobiome. So I would like to showcase to you one of very excited finding from a research group from James Cook University. So this research group, Dr. Susan is here. She also is one co-author in this paper. So one of the very excited findings in this work is this research group, they focus on genetic breeding. They don't focus on diseases or microbiome. They use the technique they call double digest restriction site-associated DNA sequencing. Or basically, this is a technique for next-gen sequencing as well. And they look into sleep, they look into genotyping of the host, not the pathogen. So that's the main purpose. And then the data, the unmapped sequence that they get, they look into that one and they found that, oh, there's a lot of microbiome and pathobiome there. Okay, and they have the capacity to collect samples from 4 different cohorts, including healthy fish and sick fish compared together. And this picture show you, this picture show you that the microbiome, viral microbiome and bacterial microbiome, in the healthy fish, they have the certain dominant group of viruses and bacteria. But in sick fish, it's shipped to SDDV at the major. 80-90% the sequence come up at SDDV and follow up with ISKNV and other pathogen. Okay, and this is a disease outbreak with SDDV. They confirm that SDDV, but it's very useful that you look into this one. And this one is only one time point, but if you look into time course, you will be able to see the shift, and you will be able to forecast. Okay, so from simple fin clips, you can do for 2 purposes. One, you look for genetic breeding. You compare which one is more resistant, and you also can look for microbiome to see the interaction of microbiome and pathobiome, and that one also very useful for forecast. Okay, so fin clip, why fin clip? Fin clip, I don't know that is why it's so good like that. But actually, when we look into fin clip, they have not only the external surface, they have internal tissue, many types of internal tissue, and a lot of capillaries there. So that means that there's the blood and there, and also they also have the mucus layer. So that is, the complex of so many things there, and it's useful for longitudinal study. And we also talk about environmental nucleic acid. This one, if we combine with next-gen sequencing, we will be able to review the composition of microbe dynamics in the system, on the fish body or in the, this eDNA, normally people look for water sample. So you should be able to understand the community of bacteria in the system. And if you have more data, then you have enough data, you should be able to track or see the shift of microbiome and pathobiome, and track the pathogen emerge in the system and dysbiosis as an early warning tool. And it's also enhanced, you can predict and you can have better management. And also this can detect the abundance of antimicrobial resistant genes circulating in aquaculture system. So this one is a little bit outside of this talk. Yesterday, someone mentioned about using disinfectants to kill pathogen in water, but recent study from my colleague in CTU Hong Kong, when they use ozone and then they look into microbiome after treatment, there's something very interesting, that the microbiome persists after infection. The majority, they are antimicrobial resistant microbes. Okay, so they have the system to pump out the ozone, so they become more resistant. So if you treat the water with disinfectant, somehow you select the more resistant one, ARG, the antimicrobial resistant bacteria. So be careful with our practice. And also other methods that we can use for non-lethal approach. So many people are using cell lines here. And in other animals and humans, they start to work with organoids. So from the cell line, they can develop the organs using scaffold, and they're able to develop the organ in the system. Or in the same chamber, they can have different area, mimic different tissue. So it helps to understand pathogenesis in the in vitro system. And also a recent study, they also use the stimulator GI tract Yeah. So that one, you don't need to use animal, but any kind of animal feed, you will be able to understand it in this system. For cell line, it's very useful for host pathogen interaction. It's useful as a model for vaccine and drug screening, and also for diagnostic, and for immune response in animals, and also for genetic engineering. Yesterday I talked to Dr. August. Yeah, he's working with CRISPR-Cas, and we just think that we have the control that not all commercial cells that work for the virus of interest, how we can engineer to make them susceptible to multiple viruses. The one kind of cell line, but it's universal for many viruses in aquaculture. And when we work with vaccine, one of the challenges is the cell they have immune response to protect them from infection. So sometimes we don't have enough viral load to make vaccine. But if we can knock down the genes, we will be able to make the factory very productive, effective in producing enough virus for vaccine production. So you just see like 10 to the 6 copy and 10 to the 10 or 10 to 11 copies. That is significant difference, million times different. Yeah. And for organoids, okay, this one is currently not really dominant, or it's not the focus of aquaculture researcher, but it can be something potential for those who have experience in cell line culture. So if you be able to make have the scaffold and make the fold in different organs that you need, it's a useful tool to study. And there are several research groups, they're starting to make some research in fish as well. And it's most excited is someone work with nutrition. So a research group from what university? Glasgow University. They be able to produce samusim. This is a system imitate, stomach, pylorexicum, and midgut, including microbe. So they put on into different, bioreactor. This reactor is for stomach, and the other reactor is for pylorexicum, and the other reactor is for midgut. And they eat as a microbe inside, and they mimic, all things similar to GI tract of salmon. And they did a research, they compare between real salmon and the system they created. And they found something very similar in like more than 98% similarity in the microbe pattern, microbiome. So it's a very promising system that can, simulate absorption, digestion of animal feed. And it also can be tested for different like probiotic, prebiotic, and the model to see how ARG gene microbes spread in the system, in the GI tract as well. And it's important is it's allied with welfare. We can do a lot of things with our animals before we go to animals. Yeah. So my question, when I read this one, my question is, can we create something like this for fish and for shrimp? With AI, with recent AI, with the 3D printing, this is possible. Yeah, you just mimic the system like that, and then you don't need to use animal, but you're able to understand, access different kind of feed, how it works, how it's absorbed, and how it's promote the microbes. You understand how the microbes, they, interact in the system. And I would like to bring you to an attention that how this method, how non-lethal method align with global fin-feed health priorities. So in February 2025, there's a workshop organized by WUA together with International Consortium for Animal Health. They bring, they have the global survey, and then they also bring experts, representative experts from different countries to have a discussion about what is a priority for fin-feed health. And I think they also on the process, or maybe already, for shrimp health as well, for crustacean health. And this result of this discussion also come up at one of policy brief And I put it, the link here. I encourage you to go and read about that one. So that may be helpful for the research direction. But I just want to bring some priorities that aligns with non-lethal approach here. That you will see that for different areas, including epidemiology, for control strategy, for diagnostic, for vaccine, and for therapeutic. For example, they said that validation of diagnostic for notifiable diseases and new diagnostic system, including e-nucleic acids and biomarker. So that is something that we about non-lethal. And also increase understanding of host pathogen environment interaction. So this is something about microbiome, pathobiome as well. Yeah, we can, normally people focus on two dimensions, that host pathogen interaction, but that not reflect the true reality. So reality, you need to put three together, host pathogen and environment interaction. And they also encourage the use of non-invasive, non-lethal sampling method. How to optimize the procedure, the protocol for this one. And for vaccine development, they also bring up the boost of mucosal immune response. Mucosal immune response is something about non-invasive vaccination strategy. Will you immersion or will you oral vaccine, and how to understand the mucosal immune system. And if we have better understanding of this one, we'll be able to develop a lot of vaccine with non-invasive strategy. And for therapeutic, they also mentioned about the method to standardization for quantification of water environment and how to reduce pathobiome. Okay, how to reduce pathobiome is critical. So if we don't have the pathobiome over the threshold, if we're able to set the threshold to predict and to check it, it's useful for disease forecast. So I would like to come with the final remarks. Ethical principle, particularly for role rooted from spiritual traditions, recognize all animal sentient being as valuing life and seeking to avoid suffering. So we are reminded to act with compassion and mindfulness. Okay, we still can do good science without killing them. And the shift from lethal to non-lethal approach, that is something have a lot of challenges in terms of practical and technical challenge. But I strongly believe that is achievable. When we we have open mind. Okay, the open mind here, sometimes we need to unlearn some old habit. There's something that we know we work very well in the lab. We have the system. We're confident with that. Sometimes you need to delete that one and to think about something beyond that one, and to move beyond traditional standard and created a new scientific pathway. Okay, for example, vaccination. Currently, when we access efficacy of vaccine, we need to use challenge tests. You give the animals like millions or billions of cells of bacteria or viruses, and that's never happened in the reality. Okay, it never happened in reality. And when you bring the vaccine to the field, it's totally different environment as well. They expose to so many things, not only the pathogen that you expose. So that is something we need to change. And we need to develop the new system to evaluate the efficacy of vaccine, not go with the system that have been set up for many years, so many years. Or the use of animal for experiment. If you go to other industry, other sector, they use very less number of animal. But for aquaculture, we multiply 10. Okay. For example, for mouse, they use only 2 animals per cage, or 1 animal per cage, and multiply 3 replicate or 2 replicate. But for us, we use 10, 20 animals per tank, and multiply 2 or 3 replicates. Yeah. So we consider the unit is the animal, not the tank. Okay, so if we're able to do so, we reduce a lot of animal, and we'll be able to understand better. And for example, when you do challenge tests, you challenge 10 feet or 20 feet with a pathogen in one tank, multiply 3. Okay, and number of samples that you collect and analyze representative 3 feet per tank. Then how about the other one? You certify them to just know the graphs. Okay, so that is something I think that we should change.
Thank you.
And don't need to method give me one more second. In advanced science, aligned with global FinFit health priority and respect animal life, and it's important, the compassion-driven research, I believe, will set a new standard where science of life, not sacrifice. Yeah, thank you very much. Yeah.
Thank you, Dr. Dong. Thank you very much. I'm afraid we won't have time for questions because there will be many questions. Yeah. I have a long list also. Yeah, okay. So I think at tea time we can catch up with Dr. Dong. Yeah. It has been an excellent presentation. Thank you very much on behalf of the society. Thank you. And now another round of excitement awaits us as we have one of the leading figures in the field of aquatic animal disease, be it diagnostics or prophylactics or treatment. Dr. Rajendran Kuluth Valapil, he is there to today enlighten us. It would be only in the fitness of things that for those who do not know him, I think most people know him, but it would be important to understand a little about his background. He just retired, but he can never retire. He's former principal scientist and head at the ICR, Central Institute of Fisheries Education, Mumbai, and a former professor at the Central University of Kerala for a stint of about 2 years. He has been a recipient of several prestigious fellowships from COSEF, CSIRO, and the Fulbright Nehru Fellowship, and has been leading pioneering research on fish and shrimp diseases, reporting several key pathogens for the first time in India, and many of them even joining hands with others and reporting for the first time globally. He has over 130 peer-reviewed publications and contributes as editor of virus disease and also as editorial board member of fish and shellfish immunology reports. Currently, he also serves as adjunct professor at the Central Institute of Fisheries Education, Mumbai. On a personal note, in my opinion, he is one of the best scientists I have known, interacted, someone who works not just doing science, but doing science very ethically, publishing, holding hands, and cooperating with many, many people. Please give him a big hand. Yeah. I'm happy to present Dr. Rajendran.
Thank you so much, madam, for your kind words. Good morning, everyone. I'm delighted to present a kind of story, a decadal story on EHP in Indian shrimp aquaculture. So it's essentially a combined presentation. Actually, the highlights of research conducted by ICR, Central Institute of Fisheries Education, and CIBA Chennai. Thanks to Dr. Kuldeep for kind invitation, and also kind of nudging me to give a presentation on this topic. So it's a long story. Actually, I try to make it as short as I can. However, this is what the introduction is. So before beginning the story, I would like to contextualize this talk or contextualize that story to look at the disease progression or evolution of diseases happening in shrimp aquaculture. We have started, or we have had viral pandemics in early 90s and followed it with bacterial pandemics, AHP&D in Asian countries, many of the Asian countries. And then from there, we evolved to, or we witnessed the chronic disease condition caused by EHP. So that's a kind of, an evolution happened during past 3 decades or so. And why I brought this slide is actually, this is an excellent example of how diseases actually defines the contours of aquaculture research. Actually, essentially, it is dictating the story. So what essentially I'm going to tell you is actually a story dictated by EHP during the past decade in Indian shrimp aquaculture. So the story started in 2016. We first reported EHP from Indian shrimp aquaculture, Venamai aquaculture from the east coast of India. And we used a full complement of diagnostic tools. And also, I would like to make parallel with the white spot history. Actually, when white spot came in 1992, we didn't have the wherewithal to actually implement a diagnostic capability. So it took almost 3 years for us to actually identify the disease and reporting it. But if you look at the today's scenario in 2016 when EHP came in, we have all the batteries of research tools available, and we were able to diagnose the disease and report it immediately. And you can see here, it's a first report from India, and also high prevalence of this particular pathogen, particular parasites, and also high intensity of infection. Along with that, we had correlation, the occurrence of white feces syndrome and also the slow growth. However, we couldn't find any statistical correlation between the presence of EHP and all those syndromic conditions. So the story started in 2016. It's almost a decade now. So the story is actually where we stand today. And it's almost like a dovetailing yesterday's presentation, a wonderful presentation given by Dr. Kalaya on the story of EHP from Thailand. So this will almost complement that. Some of there'll be overlap, and some new information will be provided. So from 2016, I thought, I'll just look at white spot again, because during the first decade, what was the story of white spot? You can see large number of publications coming in different journals. When you compare with the white spot syndrome virus story, EHP story was slightly different because we have little less publications compared to white spot. It's because of obvious reason, because white spot was a kind of explosive epidemic, and several people were actually investigating that disease. However, when it comes to EHP, the number of people who are investigating that disease is not very high, and also it's a chronic, a subtle condition. So probably that may be the reasons why the publication rate. And if you compare even the Indian publication with international publication, we have a limited number of publications. But if you look at critically the research agenda or the research thrust areas which we have followed in the case of white spot was almost parallel, almost similar to what we followed with the EHP. All those the thrust areas which we followed for white spot is almost similar. So if you look at the distribution and the spread of the EHP, 2016, you can see here, it was completely reported, completely restricted to east coast of India. Again, there was obvious reason for this, because the coast is the epicenter of shrimp aquaculture in India, and also people were only looking at the shrimp cultivated in that area. And when you look at 2021 onwards, we have the distribution and spread across the west coast of India. And significantly and interestingly, so this is a very significant observation where EHP is being reported from inland saline areas in the northern states of India. And why it is interesting and significant, that's considered to be a somewhat biosecure area compared to the coastal areas in the east and west coast. So this is actually, we do not know exactly the pathway of incursion of this disease into those inland area. Probably it may be through the infected post larvae, and it's only an assumption. So in this research highlights, you can see large number of carrier hosts actually reported of carrying EHP infection. Even a range of crustaceans, molluscans, even aquatic insects. And also recently we have found that the freshwater prawn, Macrobacterium rosenberg, is also carrying an infection. However, I take it with a pinch of salt because this is essentially based on only PCR screening. And also the PCR screening, also some of the reports are actually PCR done with the non-specific 18S SSURNA PCR, which may not be a reliable source. And also, we need to really relook into this because it's very important. The carrier host potential actually determines the epidemiology. So we need to really critically relook into this, what will be the farm level consequence of this information, and also what is the clear potential of these intermediate hosts or the carrier host in the transmission of EHP. So now the story is actually, yesterday we had a question, what is the status of P. monoron? This is one of the students' work. All these things are actually students' work. So you can see here, we have a comprehensive survey on the incidence or the prevalence of EHP in India, and we covered both the East Coast and West Coast. A large number of samples have been analyzed for the presence of EHP. You can see here a high prevalence, but reasonably less than what we have observed in the case of Penes venami. And also one of the interesting things is actually the intensity of infection. We have a low level of infection compared to P. venami. So that's a kind of, so the context of this particular investigation is that the Aquaculture Authority in India, where they were trying to reintroduce the tiger shrimp back into the culture situation, considering the fact that venami was very, very susceptible to this particular disease. So then we went on to, actually what we have done is, it's only a wild, kind of, information available in the distribution. And also one of the interesting things in the survey is, only one of the samples, throughout India we collected hundreds of samples. Out of only one samples came up positive in the first step of PCR. That means we had a very low level of infection in monodon. So what we did is actually we went on to look at the susceptibility. So we conducted a comparative analysis or comparative susceptibility study comparing the susceptibility of monodon with Penes venami. And we did a range of tools employed to see the susceptibility. So first of all, we identified, or we just looked at the histopathology, the PCR or the EHP load in the animals, and also in situ hybridization, and also growth parameters. And what we have observed is actually EHP in Penes monodon, we have a low level of infection. The EHP load was substantially low compared to venami. And also monodon was kind of showing a kind of resistance. It's not a resistance, actually a kind of tolerance. And you can see significant difference in the specific growth rate and average daily growth in the case of the venami. And also higher survival of monodon compared to venami. So this indicates, or it's a kind of evidence to show that Penes monodon is comparatively resilient to this particular pathogen. So then we took it a little further. We looked at the microbial profile of the monodon in comparison with the venami, and we found that the microbial dysbiosis caused by EHP is substantially or significantly little or significantly less than what we have observed in the case of venami. And then we looked at the transcriptome profile in the infected hepatopancreas, where we found that the regulation, the transcriptome profile also was slightly different from what we have observed, what it was reported in Penes venami. Then you can see a parallel study done by SIBA on the susceptibility of Penes venami. You can see a large number of variables looked at: immunological profile, the metabolic profiles, and the digestive enzyme, and also growth parameters. And what you can see is here, the EHP is significantly affecting the metabolism, lipid profile, lipid metabolism, the shrimp physiology altogether, and growth and the immune responses. So it has got a tremendous impact, or a significant impact on Penes venami when you compare it with the monodon. So then we also identified some of the risk factors associated in the farm outbreak. You can see more or less similar risk factors identified, especially if you have a disease outbreak in the previous farming practice, then you have more likelihood of getting the disease in the subsequent condition. And also in the case of monodon, in some of the farms, they use wild stock because they don't go for SPF, wild stock. So when there is a wild seed used for stocking, you have all the chance of getting EHP into the system. So these are all some of the critical risk factors associated with the EHP infection in farming situation. Coming to some of the interesting studies which carried out by SIBA, an in situ, a kind of a germination study under the scanning electron microscopy. So this information will actually help in studying the biology and the transmission of EHP. And then they also actually generated the whole genome data, the whole genome sequence of EHP in India, and it is found that it is almost 98 to 99% similar with the other isolates from China, Taiwan, and other countries. So then they also developed a field deployable diagnostic tool based on the LAMP technique, and it is highly sensitive, highly rapid, and also it has been validated in the field. So this is a very significant, or a very kind of field deployable tool for easy and rapid diagnosis. So then yesterday we had a kind of lot of discussion on the occurrence of white feces syndrome. So what this particular study, what is demonstrated is EHP is one of the contributing factors of developing white feces syndrome in association with probably cohabiting bacterial pathogens. You can see here, if you feed or if you use a challenge experiment with infected hepatopancreas, you get a white feces, the transformed microvilli, and the white feces in the culture ponds. Whereas if you just use the EHP purified spores, it will not generate white feces as such, but it can generate white guts in the shrimp. However, in the control, you can see neat and clean, squeaky clean, kind of, not a white incidence of whitish discoloration. So this is what it is demonstrated is that there is a co-infecting pathogen, but EHP is the contributing factor for the development of white feces. So then, again, I think Dr. Kalaya has put it, evidence for the vertical transmission. This actually supporting this study also support the postulate that it is highly unlikely that EHP can get transmitted vertically, especially transovarially. So the whole transmission will be horizontal through cannibalism and spores. So this is a kind of evidence to prove that. So then we have the evaluation of disinfection, a range of chemicals which can be used for disinfecting farm appliances, and also some of the chemicals which can be used for aquaculture environment like water and sediments. So this also demonstrated by SIBA to look at, by selecting some of the important chemicals. So this can be a kind of disinfecting agent which can be used with evidence. So then this is an interesting study or an important study. What we have done is actually docking some of the parasite proteins with available drug molecules and also plant-derived molecules. And we found large number of parasite proteins which has got a docking potential. And that can be actually docked against several of the drug molecules as well as the plant molecules. And the take-home message is actually there are several molecules, EHP virulence proteins, that are actually suitable for docking studies. And also, we identified drug molecules which can have a thorough docking, and that can be a potential therapeutic agent. You can see here commercial drug molecule. Of course, it may not be a proposition to apply in aquaculture scenario, but the bottom line is if you can use the plant-derived material which can be incorporated in the feed, that will be a very useful therapeutic agent. And taking this message forward, or taking this lesson cue forward, SIBA has come up with an excellent product. I think it is available on the website. So it's called a SIBA EHP Cura Grow Plus. It's a phytobiotic formulation based on the molecular docking study. And you can see they essentially looked at all the components like the enhanced immunity, inhibition of spore germination, its reduction in the EHP load, and of course the growth parameters which they looked at. And it is a highly effective formulation. You can see EHP load is getting substantially reduced, and also average daily growth is substantially increased, and also it's economically viable and a kind of suitable therapeutic agent which can be incorporated in the feed and applied in the farm very effectively. So I'm just trying to summarize. As I mentioned, the whole it's actually a kind of fast-paced story which I narrated because of time constraint. You can see here we have started the journey in 2016 with the first report, and spread in the west. East Coast was the first, the area where we reported, and it spread to the west coast and even to the inland areas in the northern India. And we almost get almost more than 35 research publications in impactful journals. I just avoided most of the journals which are not having an impactful, impact journals. And also a range of potential carriers we have identified. And as I mentioned, I again underline the fact that it has to be relooked carefully before we actually get a farm level consequence for that. And also prevalence and distribution of P. mondon, that's very essential because the Indian government or Indian aquaculture is trying to reintroduce the native species mondon. So it's a silver line in aquaculture where we can think of a comparatively tolerant species as an alternative species to Venami. and also key farm risk factors which can be employed or which can be integrated in the biosecurity protocol, and also artificial germination, which will help us to understand the spore germination. Whole genome sequence available now, and also sensitive field deployable diagnostics. And association of EHP and WFS, that's also an indication or experimental evidence to show that EHP is a contributing factor along with some of the other pathogens. Of course, bacteria will be the potential one. And absence of vertical transmission, that's a very good news, because if the vertical transmission is possible, then it's a serious problem in controlling the disease, serious issue. And also potential therapeutic molecules we identified, and an effective phytobiotic is available right now. So this is actually the snapshot of what we have. We have traveled the whole story dictated by EHP during the past decade, 2016 to 2026. So this is where we stand today. So before I conclude, I would like to give some suggested areas or potential areas which we need to look into. One of the areas is actually we had to decipher the host pathogen interaction critically at the molecular level, because that gives us more confidence on how EHP is actually affecting the host. We have the phenomenal observation of growth retardation, WFS, but we don't have the mechanistic explanation to prove that, okay, this is the reason why the EHP is causing. We know, know very, very clearly that how a microsporidian affect the metabolism. But still, we have some gray areas to understand. And also, there is a potential of employing genome editing and genomic selection process. I think some of the companies came up with the genomic selection process because they found that heritability is very high, so you can really employ or you can really generate the resistant stock of shrimp. And again, coming to the diagnostics, though we have a range of diagnostics available, but still there is a gap. There is a necessity for a field employable, a kind of point of care kit where the farmers can use it at the farm level, and it can, they can take a farm level decision on the spot. So that gap is still existing. And also phytobiotic, which I had just explained, which is developed by SIBA. Actually, there is still scope for enlarging that scope and also looking at some more plant product or some plant derivatives for effective in developing therapeutic solution. Having said that, what I would argue is actually, apart from the deep science-based solutions, I think we need to be very practical. We need to be looking into other aspects like biosecurity, because we need to actually re-look into the biosecurity protocols which we follow currently on EHP, considering the fact that the EHP spores have a low or a short viability period. And also, we have a kind of less susceptible or a tolerant species for alternating the culture period. So either a fallowing or a crop holiday followed by alternating the crop by species could be a kind of a strategic biosecurity protocol which can be followed to actually circumvent EHP infection. But the issue is actually it is easier than done, because biosecurity is a kind of tricky proposition to be kind of implemented at the farm level, because we have majority of small-scale farmers. They don't have the wherewithal to implement a strong biosecurity protocol in place. So what we need to do is actually we have to incentivize that process. What I suggest is actually there should be a biosecurity-linked insurance policy, because insurance should be strictly compliant with the biosecurity which is implemented at the farm level. So that will incentivize the farmers to actually look into a better biosecurity protocol. Probably that will be the way forward to actually looking beyond the science-based solutions for EHP. Probably it may be applicable to other diseases as well. So before I signing off, I would actually profusely thank Dr. Satish. Yeah, he's a prominent EHP researcher in India right now, working with SIBA Chennai, and he was one of my students previously. And one of my PhD students, Dr. Naveen Rajeshwar, he also carried out part of his work in SIBA. I thank profusely Dr. Kuldeep, because he facilitated the whole process of even funding some of the work which we carried out on Penaeus monodon. Thank you, Dr. Kuldeep. I really appreciate your effort, and also nudging me to kind of come forward to give this presentation. And I also thank the august audience, the distinguished delegates for your valuable time and patient hearing. Thank you so much for your time.
Thank you. Thank you very much, Dr. Rajendran, for that journey.